All right, we're drawing. All right. So we're talking about cellular things. Ignore that circle. And what I want us to be able to imagine is all these things that we've talked about occurring in any given cell at any given time, right? And namely for us, that's a plant cell. Just to throw out some terminology from past classes, it could be a parenchyma cell or a sclerenchyma cell. It could be cambium. It could be a pluripotent cell. It could be any cell from any organelle or organ in a plant, okay? All cells undergo things that we've talked about today. Well, I take that back. When we talk about meiosis, that's only the gametophytes, right? The megasporophyte and microsporophyte. So I'm gonna go ahead and draw right here, cell, and its typical nucleus maybe. I'm gonna make it real dark just because these are all strands of DNA, right? And you have some large central vacuole, you have ER, and you have all those organelles in there. So what I'm going to do is talk about the what you've viewed as the life cycle of a cell. So I'm going to put a circle right about here, and I drew this on the whiteboard back when we first covered it. And you might remember, you've seen this possibly in other textbooks. And sometimes you'll see this divided with another little one right here for the C. That's a C. All right, and this is the whole entirety of the life of a cell. Okay, 100% of its life from we use the word birth to reproduction. So I'm going to do right here is I'm going to draw the cell about this big. These are the cell walls, and I'm just going to go ahead and focus on the nucleus. So here is the nucleus, and I'm going to put 4 chromosomes in that nucleus. Oh, you know what? Let me make this bigger over here. I don't know if that helps. Yes. And I can also zoom in a little. Well, we'll just leave it like that for now. If you need it bigger, I can make it bigger. I can zoom in. That's a C, not an L. Okay, so I'm going to put 1, 2, 3, 4 strands of DNA in there. And when we apply stains to cells, that nucleus stains extra dark because it has a lot of nucleic acid, a lot of protein, nucleoprotein. And that's about, let's just say, then the size. And through its life, all right, through the G1, now I'm not going to have you memorize this, it's just to give you a mental picture, right? This is the majority of the life of the cell right here in G1, the first gap stage, or as I like to call it, the first growth stage. It's maybe becoming specialized and differentiating to do the functions it's designed to do for that particular cell. Whatever it is, I generally refer to it as ADL, activities of daily living, right? Whatever proteins that cell needs to make, it's doing it. Now, how does it do that? That's the topic of today's lecture, but this is a quick review, which I forgot I wanted to time myself because I don't want to go too long on this quick review. I will. All right, so that cell grows, and so that's my attempt to make it just a little bigger. Does it look a little bigger? And the nucleus is about the same size, 1, 2, 3, 4 chromosomes in it. And the cell grows, but there is a cell size limit. Cells tend to be small for good reason. And so the signal comes, hey, it's time to reproduce, or you're going to reproduce. Let's just call this a meristem cell. Remember the meristematic regions are regions where there's perpetual growth or whatnot, or even in the, again, the reproductive structures of the flower. We could call one of these cells pluripotent cell. So whatever the message is, it comes from the outside, whether that's physical pressure or whether that's a hormonal messenger. But the message is, hey, it's time to reproduce. Reproduction. And so it gets that message, and things start changing all of a sudden. The physiology of that cell starts to change. Certain parts of the DNA start to get wound up, and certain parts get more strung out. And these are now going to be proteins, and most of the energy of that cell is going to go to one primary function. I mean, of course, life functions still need to happen, but the primary thing is then DNA replication. So where we used to have 4, 1, 2, 3, oh, that's a long one. 4, that's a short one. So we have 1, 2, 3, 4 different, what you would call maybe chromosomes in the structure of chromatin during the life of the cell. So at this point, the DNA, as you know it, right, a double helix. We're going to go ahead and just draw this kind of a thick line here to represent the double helix, because it'd be a pain to try to understand this drawing looking at it at this level. But generally speaking, this DNA exists as chromatin, all kind of, whoops, sorry, hold on. Hope you're not right on target with me. 1, 2, 1, 2, you know, just trying to be 1, 2, 1, 2, accurate here. And what we have in here are, I don't know if you've ever heard of hemoglobin being having quaternary structure. We have 4 units or substructures that make up this structure called the histone. And this is a protein. These are histone proteins. And the DNA is actually wrapped around these histone proteins, in large part because not all the DNA is needed in all the cells all the time. So a lot of it is just kind of packaged up in spools like this into these structures that we generally call nucleosomes. All right, but even this is a little too close still. And I'm going to go ahead and make it a thinner line over here. And what we generally see, and this is actually about the magnification we'll look at highest under a microscope. We call it kind of beads on a string. But I want you to understand that this is the structure that we refer to as chromatin. That N refers to kind of a protein structure. Chroma means colored bodies or colored proteins. It's really DNA wrapped around histones, but it retained that name from way back when, before they knew what it exactly was. So you have DNA wrapped around these histones that form structures called nucleosomes, or commonly referred to as beads on a string. And so you have these DNA strands, and you understand this is the double helix, and this is where it's wrapped. And generally then it can fall into one of 2 categories. Now you'll find some sections that have a lot of histone and supercoiled. And so you can have euchromatin, and you can have heterochromatin. And the difference is, one is accessible and willing and able to be used, able to be read, able to be expressed. The other tends to be, such as in this case right here, maybe all the DNA that's wrapped around these spools, I mean, it's tightly wrapped around those spools. Generally speaking, it's not accessible for use. Can't express itself, we can't get the message. It's just been stored away. It no longer needs those genes. You know, you only need to make fingers once, right? Once those genes and proteins, or the dying back maybe, whatever it was, don't need them anymore, for a simplified example. That'd be heterochromatin, generally speaking. So as chromatin exists, or DNA, I'll just go ahead and label this, DNA exists in this manner throughout G1, S, and G2, okay? So chromatin is a structure of DNA during this time. So if we can imagine, another thing we covered then was once we hit S phase, does anybody remember what S phase is? You see that? Interphase had 3 phases, G1, S, and G2. And then we had, what we're going to go ahead and do is meiosis. And then maybe cytokinesis. In meiosis, we had prophase 1, metaphase 1.
We can't see what you're writing.
What's that?
We cannot see what you're writing.
Oh, okay. I'll expose it here shortly. I'm just, all the stages of meiosis: prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, and then the T2 for telophase 2. So these are all the stages. Now, I'm not having you memorize. You don't need to remember all that. The only things you want to kind of know or remember is that this is where DNA is synthesized, right? We have DNA is copied here, a replication. That's important. And so we learned about helicase, splitting it, right? The replication fork, we had discontinuous, and then we had new DNA synthesized. And so there became 2 DNA where there used to be just 1 DNA, right? So we had 4 chromosomes, 1, 2, 3, 4, and that's just commonly speaking. We know it exists as a chromatin, but we're going to call them chromosomes that have this centerpiece called a centromere. And once we've gone through the S phase, the cell has probably grown a little bit more. The nucleus has probably gotten a little bit larger. And now we have 1, 1, 2, 2, 3, 3, 4, 4. Now we have double the amount of DNA. Does that make sense? Because everywhere there used to be 1, now there are 2 strands. Sisters don't like to be separated. I have a lot of friends that are twins. I'm a twin, and all the guy twins separate us. I don't care. But I know at least 3 girl twins, and they don't like to be separated. So let's call them sister chromatids. So these sister chromatids, I'm going to go ahead and just blow this up over here real quick to say that once we go through the second growing stage, the organelles also reproduce and multiply themselves, such as ER is added, the mitochondria duplicate, chloroplasts duplicate. So it's going to be a lot bigger of a cell here in order to handle that. And again, 1, 1, 2, 2, 3, 3, 4, 4. So we still got a lot of DNA in there. And what's going to happen, I didn't bring my bags, I should have got those bags, is you're going to go from a whole lot of, just imagine a big ball of string, just a mess of string. I mean, if you've ever had I don't know if you yarn or knit, but I imagine my grandmother knitting in a basket right next, and they're in these nice, well-wrapped spools or balls. That's why they sell it. You don't go to Walmart or wherever and just start picking up and measuring yarn by the yard, right? And wad it up and throw it in a bag. Gets tangled up, it's unorganized, you don't know how to. So DNA has to organize itself, and it further organizes by continuing to wrap around those histones, but also coiling and then supercoiling. And so if I were to draw this DNA out here, we got a lot of histones in the centromere region. And so that would have been before duplication. Oh, too bad. But now that we're here, where we've duplicated it, what was 1 chromosome with the centromere is now going to be 2 sister chromosomes or chromatids held by centromere. So we need another one. And they don't want to separate, and those proteins hold them together. And you get the idea that's a really long strand. And this is generally what we call a duplicated chromosome. How did it become duplicated? DNA replication in a semi-conservative fashion. Those terms make sense, rejog your memory. So you have semi-conservative replication, where each of these DNA strands, actually sister chromatids, have one old and one new, or one original and one newly synthesized strand. So these sister chromatids make up the duplicated chromosome. And meiosis is then all about, I'm not going to draw all the sequences of meiosis, but we know we had, again, the prophase, and metaphase, anaphase, telophase, and to squeeze it in here, again, prophase, metaphase, anaphase, telophase. You get the idea, hopefully. Such that by the end, you're going to have four, right, brand new. In this case, because it's meiosis I usually do this with mitosis, but it can be used to illustrate meiosis as well. But you're going to have four haploid. Of course, this goes on to fertilization and then grows and enters into its own cell cycle. So what we've covered this semester so far is an amount about the structure of DNA and its base pairing rules. A binds to T, G binds to C. There's hydrogen bonds that hold the two together. I didn't really go into histones previous to today, but that DNA exists in the form of chromatin in your cells, the euchromatin and heterochromatin. When the time comes to replicate, we call that DNA replication. Oops. Sorry. And it occurs at a replication fork. We had the helicase enzyme. We had primase. We had DNA polymerase to make another polymer of DNA. And we had, what else? Ligase to link the phosphodiester bonds, and so forth. We learned about the endonucleases and the exonucleases, which can snip and repair if there are mistakes during the S phase. And so we learned just a little bit of DNA repair through base excision repair or nucleotide excision repair, NUR and BUR, mismatch repair. Again, no details, but just the general idea of how it proofreads itself. We learned about the direction. We read up, right? So we go from a 3, it's read in a 3' to 5', while synthesized in a 5' to 3' direction. So those are minor, well, important details, but none that I'm going to draw here. But you won't be able to draw that, and we can practice that specific drawing later. And more recently, in the last 2 weeks, we've learned or covered very important concepts here, namely crossing over between non-sister chromatids of homologous chromosomes. So if there's a gene in this segment of DNA, right, then that same gene's got to be over here too, because it's a replica. But that's only one of your chromosomes, right? The other homologous chromosome, let's just do this, is there also. So that one might have been AA, but the other one might have been a different allele, a little a, little a. And so in meiosis, these two homologous chromosomes, homologous because they code for the same trait, are able to cross over and exchange parts. That's a big deal. That's where we get recombination. And that's the power of sexual reproduction. New genotypes, recombination. We learned that, of course, reducing the chromosome number from diploid to haploid is important. And that happens here as these homologous chromosomes line up. So homologous chromosomes, you can write it, I didn't leave myself enough room. Homologous chromosomes line up, and homologous chromosomes separate. And that's big. And this is what reduces our numbers from diploid to being haploid. But it's also what we want to know for when that doesn't happen, it's also how we can get various ploidy numbers. Whenever this fails to occur and we go through the process and we begin to duplicate the genome, then we get 2 sets of chromosomes, which that part I will leave for breeding next term, right? But we covered as much as the cell cycle, replication to understand when and why and how it occurs, and then meiosis to understand 2 concepts. One, the concept of recombination, crossing over. The paper I gave y'all dealt with some normal and mutant proteins that work like topoisomerases in order to SNP. So those are the molecular biology components of how this DNA gets cut and this DNA gets cut. And I was telling Mary yesterday, you know, we like to draw these pictures of the DNA arms, just kind of entangling each other and pulling apart magically, swapping parts. It's not at all like that. They come up close to each other, kind of a web of proteins. Anyway, proteins hold them together, and these specialized proteins you've read about, hopefully, snipped them, moved them, and then another enzyme came in and annealed them. And what you have is an exchange of reciprocal parts of the chromosome. And this is not what we're going to go into, and you don't have to remember, but just to say the application is very interesting because this is where sometimes mutations and chromosomal duplications can occur. Or if it gets flipped upside down, you can have an inversion. Those are mutations. So the paper that I gave on meiosis had to do with a little more of the details of the crossing over, how that is done. And of course, you probably had to look up a lot of stuff. Hopefully you found some pictures that helped you understand it. And then the second part of that was the ploiding, which we may get into or refer to again later in the semester. All right, so that's more or less a review of the topics we've covered, somewhat of a big picture up until this point in time. I'm sure as a review, I left out certain detail, but are there any questions on the scribbles you see up here? Spread that out. About that for something else. Or going up there and Karen, any questions?
I don't think so.
Okay. Let's now go to what do you see now?
The PowerPoint.
Perfect. Oh, what slide was it? 28. So see the PowerPoint?
Yep.
Okay. So with that, so we had the how to read a paper. Hopefully, I think you probably, y'all learn a little bit from that, just from answering those questions. I mean, we learned a little bit about flower color and one way in which different flower colors can be produced in the same plant. So you'll be getting more papers probably not until after the midterm. I had one for today, but once I decided to review, I knew we wouldn't get into the small RNA stuff. So I might post it as supplementary just for fun. We'll see. Okay. Y'all like that picture? Y'all know who she is? Yeah. All right. Can you see? Can you tell the single stranded from the double stranded? So as an overview, this is what we're doing today. We're kind of going through this process. If you recall DNA replication, then this is not going to be too much more difficult because it's very similar. If we use DNA polymerase to make more DNA, then what do we use to make RNA? RNA polymerase. Okay. So there's 3 kinds of RNA. And all I'm doing, Karen, is writing 3 kinds of RNA. And so guess what? Well, let me go ahead and write it. So there's ribosomal RNA. There's messenger RNA. And there's transfer RNA. And there are 3 kinds of RNA polymerases. Okay. Each kind of RNA actually has its own polymerase. So our RNA is made, so that's a single strand of DNA, right? That's the single stranded. And that's made by RNA polymerase 1. Okay. Messenger RNA, mRNA, is made by RNA polymerase 2. And tRNA is made by RNA polymerase 3. Do you remember that? Thrice, tri, 3, T, tRNA, 3 polymerase RNA version. Anyway. So this is generally what an mRNA looks like. It's probably easiest to imagine because it's just a single-stranded polynucleotide, very similar to DNA, but single-stranded. Ribosomal RNA is the goofiest looking one. They got all kinds of structures, but generally speaking, that's usually coupled with a protein, though, to perform its function. And then transfer RNA. Don't worry, we'll get to this. This is the same PowerPoint that I gave you. I just threw these 4 slides in there right before the lecture. And I will put this one in place of that one because I also have 2 video links in it. But it is the same lecture. Transfer RNA, the third kind, generally looks like this. This is what it looks like if you were to take this middle one, and this is not the best picture, but if you took this blue portion and this orange portion and pulled them apart, you know, it's kind of like this. But when you open it up, it looks like this clover here, okay? But these are kind of folded. You can see the orange and the yellow. So there's various ways you'll see it's represented in books and in PowerPoints. They are all accurate representations, but it can be confusing because in the end we say, how does that look like this? Well, this was just a flat 2-dimensional version, but in reality, it's folded, twisted over, and as a cartoon picture, I guess. There's also a space filling model we use too, and a ribbon model. Well, that is a ribbon model. But so we won't see this one again until next week. So let's go to the transcription. And this is the beginning of how we make a protein. Y'all can read that while I catch my breath.
Where's this PowerPoint at?
It should be the one, I think I posted it last week under Unit 5.
Next slide.
Well, which slide are you
So I can find one over writing.
No, so
What is it called then?
under
I
Blackboard,
have it, yeah.
yeah. So it's this one, slides 1 through 27. I'm on slides 20. So just before lecture,
okay, so those are the
I just added her and these 3 slides and that picture. It's the same slide, just additional picture. Actually, I'm going to do, oh, I can't. Is that where it was? Yep. So if you can see back there in the cheat sheets, the only difference between thymine and uracil is one functional group, and that's methionine, or methyl group, I should say, the CH3. Otherwise, when you look at the structure of this, all the bonding patterns, the covalent, single, and double, identical. So that's why it's able to pull a swap. And it's good that it recognizes that difference. I tell you, again, in biochemistry or microbiology, knowing just the power of those functional groups plays a big role if then you're wanting to understand how and why. I mean, we have big name changes in these molecules based on very small changes to the actual structure of the atom or molecule, excuse me. Yes, ma'am.
This slide is in our notes right now, but that
Picture?
diagram is not. Is it meant to be, or is it just like an add-on for this set?
Yeah, so it'll be there. I mean, as soon as this lecture is over, I'm going to take that one down, put this one up.
Okay, it just wasn't on this one, even though everything else was, so I didn't know if it was something added on or not.
Yeah. My apologies. I mean, and I'll always be editing up until the last minute as I go back and so what can I add to make it better. But I wanted y'all to have it at least through the week. You can look at it if you wanted to. This intro. So likewise, this video is thrown in there. You have the slide, but without the video. So the DNA sequence where RNA polymerase attaches is called the promoter. So there's a lot of exciting things here that I'm not mentioning. We're just kind of going about it through a ho-hum factual thing. But the question is that you all have in the back of your head when you're able to process is, how does it know to open up that DNA strand? Where does it open up that DNA strand? When does it open up that DNA strand, right? So there's a lot of regulation and control that we'll want to talk about, and we will in time. But one thing we learn is that DNA is not just these protein coding sequences. The protein coding sequences are very minimal in the genome of any organism. Even though proteins do everything, but it seems the protein coding segments of DNA were the cast. It seems the crew and the tech and the director and all the behind the scenes lighting are much, much more of the production than just a few members we see saying lines. And that's the case here. Actual coding segments are the minority, but that's what we see. This is what we test. These are the phenotypes we look at, but there's so much more going on in the regulation and in the control of those actors, those players, the proteins. So here's, again, just to form a picture. So we're going to go through several slides that are just writing and pictures. I think animations are a little more powerful. Let me do this real quick. You can click on it there when you review. But I'm going to get in there. Copy and paste this so that you can watch it first.
Transcription is the first step in the expression of the gene. It is the formation of a specific RNA sequence from a specific DNA sequence. Transcription initiation occurs at a promoter, a region on the DNA, where RNA polymerase binds. RNA polymerase attaches to the promoter and begins RNA.
Can y'all hear it well enough? I don't know where the volume is.
Sequence from a specific DNA sequence. Transcription initiation occurs at a promoter, a region of the DNA, where RNA polymerase binds. RNA polymerase attaches to the promoter and begins to unwind the DNA. At the initiation site, the polymerase begins reading the DNA template strand and building a complementary RNA strand from 3 nucleoside triphosphates.
All right. So what are two differences you see right away what unwraps or unwinds the DNA?
RNA.
There was no helicase in this. So that's one difference between DNA synthesis and RNA synthesis, is that DNA polymerases cannot or will not bind to the double strand and will not cut those hydrogen bonds, which is the job of helicase in that case. Here, now there's a lot more proteins here than what we're seeing. Again, this is an overview. But here the RNA polymerase, in this case, RNA polymerase 2, is the one doing the binding and pulling the two strands apart. The second thing you notice, what is like DNA is that it's going in the same direction, right? Writing down 5' to 3', adding 3' ends. Then I guess the third thing is only one strand is being used, right? Whereas in DNA replication, we had it going in both directions on both sides, right? Here, you only have one. So what we'll see is there's always a template strand, the sense strand, and then there's the nonsense or non-template, sense, anti-sense. Okay.
The RNA strand grows by the addition of these substrate molecules to its 3' end. This is the elongation phase of transcription.
Elongation.
Note that the bases of the nucleotides of the RNA strand form pairs with the bases in the DNA strand. Also, note that the RNA nucleotides contain the sugar ribose, which has a 2' hydroxyl group, whereas the DNA nucleotides contain the sugar deoxyribose, which lacks this hydroxyl group. RNA polymerase functions similarly to DNA polymerase. It adds nucleotides to the 3' hydroxyl group of the last nucleotide. The DNA double helix rewinds as the RNA polymerase moves through. Like the unwinding, the rewinding is an energy-requiring process that is accomplished by RNA polymerase. When RNA polymerase reaches the termination site, the RNA transcript is released from the template. The DNA rewinds completely, and the RNA polymerase dissociates from it. The DNA and RNA polymerase can then participate in other rounds of transcription.
Yes, sir.
How many subunits does RNA pol 2 have?
So that's a good point. So notice here, this is one reason I like this animation kind of, that you, while not absolutely accurate, it gives at least the impression that we know it's not going in a single base by base by base by base. It's a large protein that sits on top of the strand. And it depends, I think, I mean, it's a range of sizes, but you're talking hundreds. It could be anywhere from 20, something like 25 small segments, and some species that's probably in bacteria and such, on up to a large number of nucleotides. Is that what you're asking? How many? So
No, how many protein subunits does that protein complex that is RNA pol 2 have?
oh, how many proteins make up RNA protein?
Yeah.
Something like 7 to 9, like that. I don't remember the exact number, but yeah, it's not one protein. That's true too. So we'll see that there's other functions, recognition sites. A lot of RNA polymerase acts just in recognizing that promoter region. But in and of itself, it doesn't just start joining. Something else brings it to it. And that's what we'll see in these few slides here, as we're about to go into. 4. Wow, that was different. All right, so that's the whole picture all at once. So we saw 3 steps in that video. We saw that there is an initiation, there's an elongation, and then there's a termination. Initiation involves proteins that recognize the promoter region first. And we call those generally transcription factors. So you will learn, all these have names. They could be like TF2A, TF3, TF1, and the TF stands for transcription factor. I'm not going to be, with all my instructors and professors in my educational career have never held me to memorizing all the proteins and enzymes. The process is important. How we learned that this is what happens is important, but it's not in the scope of this class. Unfortunately, we'll still talk, hopefully, at some point about how we learn some of these things. But understanding enough detail, of course, I guess would be an objective. That's to me the continual crime and challenge of classes like these is to what level of degree. So there'll be several transcription factors, and there is a sequence to their binding. But once those first transcription factors located, and I'm sure there's a lot of those transcription factors that might be what we call constitutively expressed, meaning on a continual basis. There's other transcription factors that are not as constitutively expressed, but are regulated by some hormone. But once they are, and that message comes, they're expressed, then they have an affinity for binding to that first transcription factor. And in turn, RNA polymerase has an affinity for those. And so it's called recruitment is what it's called. It's like, hey, there's a party on the gene. And then everyone starts coming. And when they're all together, partying, and they just fly down that strand at a really quick speed. They let the synthesis begin of mRNA. So what this is showing then after initiation, well, this is the initiation. So once all those transcription factors are there, RNA polymerase can bind, and you'll notice it's a very large protein complex. And it combines the double strand, recognizes the RNA transcripts, and we have the reading. Until it gets to, in bacteria, it's pretty easy. There's a stop sequence, and once it hits that stop sequence, the whole complex disbands. In eukaryotes, it's a little more complicated and complex than that, and we'll get to a little bit of that in a bit. And so in the end, you have this RNA, what we call a transcript. It's simply a transcript because it's a rewritten code. It's all in the same language. It's in the same language of nucleic acid, except that U replaces T. The slide's just good for reminding you of the direction that it goes, 5' to 3'. And you'll notice that wherever there was, base pairing rules are followed, except that there's a uracil in place of thymine for every adenine. And I don't know if I've used the word downstream yet, but I'll start using it here now. So those are the 3 stages we just reviewed. So promoters are what we call elements. So there are sequences on the DNA, as I mentioned a while ago, that do not code for protein, right? Those are just the main players. It might be the whole reason the lighting crew exists. They might be the whole reason, stage crew exists, or the director, is to get those main actors. And the proteins are the main actors. That means there's a lot of instruction that has to go on behind the scenes that we don't see. And the same is true in the genome. And so one of those elements of DNA are called a promoter, in the promoter region. And that's what signals the initiation. There are several promoters. A really cool thing to note, not in the slides, but for your molecular biology, is that promoters for messenger RNA for all proteins are pretty much the same, more or less, across life. bacteria, plants, right? tRNA, different, but pretty much the same through all life. And you guess, if you had to think about it, if I'd given you time to think about it, you'd probably guess that because life is dependent on the same 20 amino acids that the DNA codes for. But what is then very interesting is that our RNA is species dependent. And that's pretty cool, just to think about. And because it's species dependent, there's no point in covering this is what it looks like, right? It just depends on your model organism that you're going to study. But nonetheless, the rRNA, the genes that code for rRNA sequences in the RNA world are also on the DNA, and they don't code for proteins, do they? And so just begin to create this bigger and bigger vision of everything that is on a strand of DNA. Literally, not just the mechanics for life, aka proteins, but also all the instructions for life. Just proteins that say you do this, you do that. So the completed assembly of transcription factors and RNA polymerase II bound to a promoter is what we call transcription initiation complex. In this promoter region, you have something called the Tata box. Now, in the early days, that's all we call it. The promoter is a Tata box. In bacteria, it's as simple. In plants or eukaryotes, it's generally a little more complex. So the promoter is much longer, but within that promoter, you're going to find a Tata box, particularly for the mRNA, which is what we're most interested in. So once those transcription factors recognize the Tata box, that's pretty much where it binds to, and begins to pull in the others. So here is a start point. Oops. All right. Now notice where the start point is in relation to the promoter. This is what we call upstream. Okay. So the starting point is what we call +1. Okay. This is where the actual gene begins. The first nucleotide base that begins to code for a protein is at the +1 point on the DNA strand. The promoter kind of overlaps with that because there is, and if you can see just behind that, a region of that transcript that does not code for the protein. It's called UTRs, untranslated region. Hold on to your britches, it gets better. All right, so we have these UTRs at about the +1 spot, right before the actual part of the DNA that codes for the protein. And look how long comparatively this promoter is. And within that promoter, you have this Tata box. So several transcription factors must first bind to the DNA before RNA polymerase II can do so. So here's that first one that bound. And once that one binds, these other two can come and bind on it as well. Additional transcription factors bind to the DNA along with RNA polymerase II, forming that transcription initiation complex. So here's RNA polymerase II, the leader of the show, who will not come to the party unless everyone else is going to be there, right? And if you're all there, then I'll come. And he does, and the party gets rolling right down the line. So elongation is actually probably the easy part of this. That's just the point of, and not that there's enough interesting things to say about it, but that's just the act of grabbing these RNA nucleotides and affixing them. As we saw in the video, I didn't make a big point of it, but they are kind of self-charged and they bring with them their own energy from the triphosphate aspect. So there you go, transcription initiation complex, RNA polymerase II and associated transcription factors, which first had to be there bound to the Tata box. Good stuff. So here's the elongation. We'll just run through these slides. Reading. Moves along the DNA. It untwists the double helix 10 to 20 bases at a time. This is pretty interesting because I don't know if you've ever seen, if we need to buy a model of DNA, but one loop is 10 bases. Two is 20 bases. So what we're saying is it's about 1 helix or 2 helix long. This is about just factoids, I guess, when I remember them, I like to shout them out, but that'd be about 34 nanometers wise. It's about 2 nanometers wide. Transcription progresses at a rate of 40 nucleotides per second in eukaryotes, and the gene can be transcribed simultaneously by several RNA polymerases. I would have added another picture to this. I didn't have time. So what I want you to imagine, well, maybe I'll have another picture up here. I can make that point better later. But just imagine a bunch of boxcars on railway. If each of those boxcars were an RNA polymerase, and out of each of those boxcars, you had a growing strand of whatever, a chain or a rope. So it's not like there's a strand of DNA that's open and this RNA polymerase is you get it? And how many of those transcripts are we supposed to produce? 1? 10? 100? So as soon as one boards it, another one boards it, and another one boards it, another one. So I had like 10. That seems really nice and soggy. You get the idea? So I don't have a picture up here. So you have to have just one RNA or initiation complex. As soon as it gets going down the line, another one's forming. And it gets going, another one's forming. And you might have 3 or 4 going along a single DNA strand. That in part can be regulated as well, depending on the need for that protein or that messenger. Did I bump ahead accidentally? Yeah, so that's that third bullet there. All right. Then termination. Okay, that's what I mentioned. They hit a stop signal, a stop codon, UUU, they disband. In eukaryotes, it goes longer. What do you mean longer? Past the stop, it sees the signal. I'm just going to go a little further, just to be safe, maybe. So in eukaryotes, the polymerase continues transcription after the pre-mRNA is cleaved from a growing RNA chain. The polymerase eventually falls off the DNA. Let's go ahead. Yeah, let's go ahead and take a break right there. What side was that, 42? So I'm gonna put on about a 5-minute break, at least stretch your brain, review, ask questions. Let me go ahead and stop this. Taryn, if you're still on there or not.
Yep, I'm here.
It's gonna be